Good afternoon. Thank you for joining us today for our panel, Unexpected Solutions, the Science of Problem Solving, sponsored by the College of Natural Sciences. Scientific research is the cornerstone for understanding our universe and everything in it. Starting at an early age, we learn how scientists use basic research to advance our knowledge of the natural environment and the laws that govern it. Through applied research, scientists work out unexpected solutions quickly and with tangible outcomes that have direct benefits to individuals, communities, and the planet. Today, our expert panelists will highlight examples of applied research being conducted and how these projects are expected to impact lives, communities, and future innovation, explore differences between applied research and basic research, share how industry partnerships bring professional research experience to our students, and share their own experiences in their applied research labs. But before we get started, we have a few housekeeping notes. This session is being recorded. Attendees are on mute and cameras are off. By joining today, you are consenting to participate in a recorded session. After the recording is made, it will be captioned and all registrants will be notified when it is available for future viewing. Participants are invited to submit questions for discussion through the Q&A feature, not the chat. Following the presentation, the panelists will do their best to answer as many questions as possible in the time allotted. Now, we will introduce our panelists. Our moderator today, Mark Tuominen, is Associate Dean for Research and Innovation at the College of Natural Sciences at UMass Amherst, and Professor of Physics, where he performs research in experimental condensed matter physics and nanotechnology. Dr. Tuominen's educational innovations are in the areas of research, learning, and professional development. He received a bachelor's degree in chemical engineering and a PhD in physics from the University of Minnesota, and was a postdoctoral research associate at Harvard University. Phyllis Kuhn is president and founder of Kuhn Copper Solutions and also serves as president of Lake Erie Research Institute. Prior to that, Dr. Kuhn was director of research for 14 years and microbiologist for 18 years at the Hammett Medical Center. She is a proud UMass alumna of the Department of Plant and Soil Sciences, now at Stockbridge. Dr. Kuhn has developed unique ways to use copper to kill bacteria and inactivate viruses, including a pure copper mesh face mask and copper insert that converts a standard mask into a virucidal mask. Matthew Moore is an assistant professor in the Department of Food Sciences at UMass Amherst. He received his bachelor's degree in food science from Cornell University and a PhD in food science from North Carolina State University. Dr. Moore was an ORI's postdoctoral fellow at the Centers for Disease Control and Prevention prior to joining UMass in January 2018. His research focuses on applied and environmental biology, and he is currently developing ways to better understand and control very transmissible diseases like norovirus. Dr. Moore is working on portable rapid detection techniques that can detect viruses in less than 30 minutes without the need for an electrical grid or fancy lab equipment. Welcome. Thank you for taking the time to share your expertise with us today. As a reminder for participants, you can submit questions through the Q&A feature, not the chat. We will have time at the end of the presentation for the panelists to answer questions. Dr. Tuominen, I'm going to turn it over to you so you can lead our panelists through today's discussion.
Great, thank you, Diane. And I'm happy to be part of the webinar today. Oops, hold on a second, sorry. Gotta start my video. There we go. I'll be moderating this event and leading our panelists through our discussion on applied research. Applied research and industry partnerships it encompasses drive much of our modern innovation and serve as a foundation for advances in science and technology that make our lives easier. All of us will be discussing the exciting work that's being done by alumni and current faculty and students of the College of Natural Sciences. First, a little background. The research that comes out of the College of Natural Sciences is world-renowned, and we have spearheaded many important scientific breakthroughs and innovations. CNS faculty are routinely listed among the world's leading scientific minds in Thomson Reuters surveys. In Massachusetts, Amherst ranks third in funding from the National Science Foundation after Harvard and MIT. On campus, CNS is the largest recipient of grant funding, and CNS faculty are campus leaders in the Institute for Applied Life Sciences, also known as ILS for short, a $150 million interdisciplinary and entrepreneurial research center focused on developing new products to benefit health and medicine primarily, and to translate cutting-edge applied research into a variety of commercial business opportunities. Postdoctoral researchers, graduate and undergraduate students in ILS actively engage with faculty and contribute to a wide range of relevant and interdisciplinary research. The Institute has been providing training and coaching in entrepreneurship and innovation to faculty and students who are developing applications with strong potential in a variety of markets. I want to say a few additional words about the importance of applied research to innovation and entrepreneurship. These are hugely beneficial to our students and alumni because it allows both to embark on research partnerships with industry leaders, enhance their professional networks, gain real-world lab experience, obtain funding for exciting new projects, and strengthen their ability to collaborate with leading scientific minds around the world. Okay, with that, let's turn to our panelists, Dr. Phyllis Kuhn and Dr. Matt Moore, to start our discussion. So the first question I asked to both Phyllis and Matt. Both of you have been working on very exciting projects within and outside of the university. Can you each tell us briefly about recent applied research projects that you have been involved with? And why is this exciting? Let's ask Phyllis to go first. Phyllis, you have to unmute.
Are we okay?
Yep, we're okay now. Thank you, Phyllis.
That's fine. I have to go into a little background. Is this visible?
Yep.
CuneCopperSolutions.org is my website, and the background material and the ability to order masks is on that site. Now, what our company does is we manufacture copper mesh masks, and you can see right through them, and also small copper mesh inserts that can be adhered to any standard mask, and it will render it vericidal. These products come with a little teeny patch of copper that you can put on your arm to ensure that you don't have copper sensitivity or a copper allergy. Now, one of the questions we had to ask ourselves was, how long does the copper remain in the nasal area? Because both of these products deposit copper in the nasal area. And copper kills, it's vericidal, it kills and it activates viruses and it kills pathogens. So I'm currently working with Penn State Barron, and they are assessing the length of time the copper remains in the nasal area after the mask is worn for 2 hours. This is measured by inductively coupled mass spectrometry. Now, we do have some information that indicates that the copper remains for quite a period of time. One of our users, in her own words, said, I started coming down with a cold. I wore the mask about 3 hours one day and 2 hours the next. My cold lasted 2.5 days, and I was done with it. Another, this is a physician down in Florida, was wearing it for allergies. And he wore, in his words, the mask 20 minutes in the morning and 20 minutes in the afternoon, and he had a great suppression of his allergic symptoms. So this is indirect evidence that the copper in the NP area lasts quite a while. So that's one project we are working on. The second project, which I think is very exciting, and the reason it's exciting is because of sneezing. Can you see this picture? Sneezes go out easily 3 to 5 feet. In the first 3 feet, it's heavy droplets. After that, it's sparse droplets. Now what happens is we ask ourselves, the masks that most people are using, are they water repellent? Are they water resistant? Do they wick? Do they draw bacteria and virus through them, etc.? So this is your copper mesh mask. Now, even though it's copper and you can see through it, if you put water on it from about an inch, you can't see this, but I can see it. It's not going through. And what happens is it beads up on the surface because of surface tension. And your common surgical mask does the same thing. Now, if you put a lot of pressure on that, it will go through. What we did was to take a saliva solution and put it on the tops and bottoms of 4 different types of masks. And the first one was a surgical mask. So we did it and we cultured with a swab. In the surgical mask, I think you can see the top has lots of bacteria, the bottom has nothing. The common surgical mask is a very good mask. Now this is a copper mesh mask, top and bottom. We have the same results. In other words, even though this is a mesh, we get no bacteria or virus or anything on the other side. Now the 2 other masks that we tested were very common masks. And I don't know if you can see this or not, but if you put water or anything on them, I may as well take this off. What happens is they absorb. They absorb immediately. And they actually go through to the other side. And these are what the plates look like. So you have full growth, top and bottom. And I think that becomes very important when you consider what happens when a person sneezes. So I would only recommend that you challenge your mask and put a little drop of water on them and see if they're water repellent, water resistant, or in fact they wick. So that's the other project that we're working on that I thought was very fascinating.
Thank you, Phyllis. Matt, will you tell us about your research, your recent applied research?
As mentioned, we do research on primarily foodborne viruses, one of which is norovirus. It's the leading cause of foodborne illness globally, and it's extremely transmissible. So SARS-CoV-2 is also very a transmissible respiratory virus, but there's actually the norovirus, which has been around for about 4 or 5 decades now, that's extremely transmissible. It results in millions of illnesses globally, and it's very difficult to control. And especially for very transmissible viruses, early detection that's portable and rapid is extremely important, because if you take time to send samples out to a lab to get tested, it's so transmissible that it's already spread like wildfire within different communities. So some of the work, the applied work that we do is trying to develop really rapid portable techniques to be able to detect these viruses. And so one of those techniques or projects is actually something called Nanopore Sensing. So essentially what we do is we take these proteins that are normally in bacteria, and you can actually exploit them to bind and sense viruses in the field in a portable format. So you can actually use an instrument that's basically like the size of a candy bar, and you can take it into the field and be able to detect the viruses. One of the other really novel things about this type of portable detection is that it's very rapid, so it takes under an hour. And it also has the ability to do something called subtyping. So one of the reasons that this is valuable is when you know that you have norovirus, because it's so transmissible and because it's in the community so much, being able to subtype or figure out what strain of virus or what strain of norovirus is there is incredibly important to being able to identify an outbreak, especially if it's through foods or something of that nature. Because if you have a certain subtype that you can isolate from the food or the environment, and you can also isolate it in patients, you have a lot more confidence in saying that food was actually the agent responsible for the outbreak. And to date, we don't really have any detection methods that are really truly portable that can actually do this. And so if you actually had the ability to get subtype or strain information in the field immediately in less than an hour, that would be kind of a game changer. And that's what this Nanopore technology has the capacity to do. So it's an extremely exciting collaboration that our lab has actually with the Department of Chemistry, also in CNS with Dr. Min Chen's lab. She did a lot of the fundamental research related to these nanopore proteins and how they interact with ligands. And then she's done some applied stuff with sensing sort of biomarkers related to sort of clinical applications. But what our lab has noticed is some of these properties that the Chen lab has discovered and done a lot of really good work on would actually be extremely valuable to identifying foodborne outbreaks, especially for a pathogen like norovirus, which is extremely transmissible. Another portable detection technique that we're also focusing on is something called isothermal amplification. And essentially, it's similar to what you may have heard of with SARS-CoV-2 called PCR. And essentially, this is an alternative technique that, unlike that PCR technique that you keep hearing everybody is using for SARS-CoV-2, this technique is entirely portable. And it doesn't really need an electrical grid or any clunky or fancy machinery. So you can essentially take everything that you need to detect it and basically fit it into a suitcase. And so the actual device that you can use for this is basically the size of a small textbook. You can charge it with a solar-powered battery, so it's extremely portable. And it's actually really great for in-field identification, especially of viruses that are present in the environment or in foods, because the viruses are at very low levels. And one of the advantages of this detection technique is you don't need very much virus to be able to detect it. So that's one of the other sort of applied projects that we have going on now. And both of these would be extremely valuable to detecting and taking action against viral outbreaks.
Thanks, Matt. So in both of your work, I see a nice interplay between fundamental basic research and applied research. Could you describe how your approach to applied research is different from the way you and other researchers undertake basic research? You know, what is the difference? And whoever would like to go first, Matt, you could go first.
Okay, great. Yeah, that's a great question. I think it's a continuum, and I think actually UMass is a really great setting for applied research, in that we have so many people that are so collaborative and that do everything from the basic research to us actually taking that research and really finding specific real-world applications for it. And we have a really nice mix and a really nice culture of working together across different departments and even across different colleges. That's really exciting. And just like I mentioned with Min Chen, we have a whole bunch of other projects that are, you know, mostly applied or being specifically tailored to a very specific real world application, but they're based upon some of the amazing basic research that CNS as well as UMass does.
Thank you. Phyllis?
Well, my research is predominantly applied practical research. Basic science research in this area would be looking at the molecular level of why copper is so societal to viruses and pathogens. In the applied area, we would be looking at something like oxygen tension after 2 to 4 to 8 hours of wear of a variety of different masks. So the whole application is different. We might be looking at actually clinical studies using the mask in cases of respiratory infection, coronavirus infection, flu, even red tide. And in that aspect, that would be something that we'd be very interested in looking at. And if there are physicians out there interested in clinical research, if they need access to masks, I would be more than happy to supply them, because it's an area calling for need. It really is. Thank you.
Yeah, thanks. Yeah, I relate to what both of you are saying. I started my career at 3M, and Matt, to what you were saying about the collaborative nature of applied research. The team that I was on at 3M with its product development, the thing that was very apparent is just the different skills that were needed, the interdisciplinary skills, and that's what allowed the product to really go forward and succeed. And you know, Phyllis, I understand too that something works, we want to understand, and oftentimes that's due to discovery. You know, how does it work? Why does it work at a molecular level? That's when, you know, the applied goes back to fundamental to answer these very basic questions. So I appreciate that. So I have a different type of question here. How do these kind of projects benefit students or faculty or others at CNS? And whoever would like to answer first would be fine. Matt, do you want to go first?
Sure. Yeah, it's well, also another thing, in addition to having really great collaborative faculty at UMass, I'm really impressed by the level of our students, both graduate and undergraduate, and both through CNS as well as through scientific societies, getting to participate in this research and actually get your hands on some pipettes and actually really getting into doing research, especially as an undergraduate, is a great value to training the next generation of scientists. I believe in our department here in food science, we're actually ranked #1 in the country in terms of our research program. And we actually have a lot of our undergraduates who are really enthusiastic about getting into these sort of world-leading labs. And I believe it's over nearly 70% of our undergraduates at some point get research experience in our department, which is pretty impressive, and really it puts them ahead of the game in terms of graduate school, as well as just teaching them basic, you know, experimental planning, conducting a lot of different life science techniques. Similarly for our grad students, they get to become, you know, part of this community and get to, you know, present our research at conferences where we very regularly will win awards. I think we have like at least over 5 in this past conference year so far. So it's really impressive, the students as well, and the opportunities that both UMass and our department and the PIs in our department afford them.
Thanks. Phyllis?
Well, this is interesting. Very little research, very little formal research has been done on the various types of masks and their efficacy. For instance, what's the oxygen tension level after 2, 4, or 8 hours of wear, especially with N95 masks? What about CO2 levels? Because so little research has been performed in this area, it's ripe for publication. They're very nice projects. They can be done quickly, and they're very inexpensive. So that's what I have to say.
Yeah, thank you. Yeah, you know, it's one of these things that the necessity of the situation and everybody wearing masks bring to light these issues that, you know, wearing a mask for a long time, that it's difficult to breathe, it's the oxygen and CO2 level changing in your environment, how effective they are over time. You know, this all brings it back to doing more either basic or applied science to answer these questions, and then it circles back to improvements in products. So these are very important. Dr. Kuhn, I have another question for you. You've worked for a number of years on copper as a virucidal. Are there other applications for this material that you'd like to see developed?
Well, there is an application already developed, and unfortunately it's not used, or it's not widespread. I'd like to see its expanded use in hospital settings, and Saldago published a beautiful article in 2013. And what they did was replaced all the stainless steel in 4 or 3 intensive care units with copper alloys. What they did over a 4-year period was decrease the hospital-acquired infection rate by 58%. Now that's astounding. Now thus far, only 2.35% of the medical centers in the US are taking advantage of this research and are using copper alloys. So basically, I'd like to see all the stainless steel replaced with copper alloys, much like it was back in 1983. In 83, I published an article on doorknobs, a source of nosocomial infection. And I will tell your researchers out there, do not be turned off by simple research projects because sometimes they're the most long-lasting. That single paper stimulated a host of publications over the ensuing years. There are hundreds of publications now on the cytal activity of copper, but that was the first one that demonstrated its efficacy as a self-disinfecting metal in hospital hardware. So I would encourage your researchers, please don't turn your nose up if you see something that looks very simple. It can be exquisitely important.
Yeah, thank you, Dr. Kuhn. The importance of talking to each other, academic researchers talking to folks in industry. There's a lot of interesting fundamental problems that lie in wait, and then also, the industry folks, through these partnerships get to learn of the latest developments happening in academic labs. So those conversations are really important, and I think really an important foundation of moving things forward in both applied and fundamental research.
May I make one more comment?
Yeah, please.
I would like to see the cruise ships employ copper alloys. And the other thing I'd like to see is paper money impregnated with copper fibers.
Yeah, thank you. So Matt, how does your department work with industry partners to bring projects to fruition? Another aspect of this question is, what does your process look like, and how do faculty, or the department's process, and how do faculty and students work with them?
Well, yeah, that's a great question. So our university, I believe we have a mandate as well to have an extension capacity, or something called an extension capacity. And so we work very closely, especially in New England. There's a lot of small businesses, and it's sort of, I don't think many people know this, but they have access to us as a department and to our expertise. And so our department, as well as my lab, we regularly will be talking to different industry partners on a range of things. So food science in and of itself is very interdisciplinary and applied. And so we have partners, all the way from MNMRs to some of the partners that I work with as a virus lab. We actually tend to partner a lot with antivirals and sanitizing companies, much like Phyllis is a part of. But our entire scope of a department works very closely with industry. And in fact, we've actually formalized that relationship with a strategic research alliance, where we have a number of companies that will pay a fee to have access and to come and see sort of this cutting edge research that our department does. And it's very fruitful in terms of collaboration and forming partnerships. And so it's quite a symbiotic relationship in that we can create healthier food that lasts longer and that's safer. And so I think that's one of the many positives of our department, as well as UMass and CNS, is just having an entire office that facilitates these relationships and makes sure that we get squared away with our contracts and such. So that's often what our lab will do, as well as what our department will do as well.
Yeah, thanks, Matt. That's a very important point, this synergy between academic researchers and industry. And it's one of trust. It takes time to build the relationship, but once you have that trustful relationship, folks from industry want to come back again and again for many reasons, to hear of their latest research, to to find potential future employees in the students, and also to convey to a department like yours the kind of challenges that they would like to see solved. So it's very symbiotic. I like that very much. And I should also mention that, you know, these kind of experiences is so valuable for students. You know, if you go back 20 years ago, not very many students did research, and certainly not applied research. But hearing, Matt, that your department, you know, 70% of the students are getting hands-on research, that's very important. And, you know, we're sort of as a college and a campus doubling down on experiential learning, because that's so important, that it complements the learning of the basics in the courses, and really gives context to that understanding that's learned. So it's very valuable. I appreciate that. Either one of you want to add anything at this point before we segue into the Q&A section?
No, I just I wanted to echo your comments there, Mark. It's an amazing point, and actually we're pretty excited in food science, and that Lynn McLansborough got a USDA grant to facilitate actually a formal experiential learning program for our students, where we have a lot of the industry partners that we normally work with, and we actually, I think, are leveraging the Mount Ida campus in allowing people to get internships, and actually it sets up future positions. So we have a number of students. So that's an excellent point.
Yeah, you know, it really fires the students up to know that what they're working on has potential impact in real world applications. And those relationships with people from industry, they have some context for what their future might look like working in jobs in industry. You know, they're working with you in academic labs, so they have some sense of what that looks like if they go the academic route. But for industry jobs, it's important to have those relationships. Phyllis, do you want to add anything at this time?
Yes, years ago, I did extensive consulting for a number of different people in industry. And as the research center developed at Hammett, I had to drop those consultantships. If any of your folks have consulting relationships, do keep them up, because what's happened now is I have a very nice product, but as an individual, trying to get it into industry is almost impossible. A lot of industries have statements, do not send original research to us. If you send it to us, it's ours. Mm-hmm. It's very hostile, and unless you know someone in industry, you are not getting in, and it doesn't matter what you have. So a word of caution: if you've got consultantships, even if they're you don't think you're going to be using them much, do keep them up, because they're very important. The only way to get in is knowing someone that's in the company. Otherwise, you're going to hit a blank door.
Yeah, I appreciate that. It goes back to this trusted relationship, you know, and as Matt knows, and, you know, other sort of university industry relations that we have on campus, you know, there's different levels of discussion. And once it gets down into the proprietary aspect, usually a non-disclosure agreement is signed, and the university's Innovation Institute helps with the agreements so that people can talk, you know, scientist to scientist, from university to industry, and have things go forward. So that's important, but it does work out. You know, we do work these things out, because in the college and in the campus, we have a lot of these industry relationships now, and it's very productive and synergistic. So why don't we pivot to some of the questions that have been sent in, and you can feel free to send questions in through the Q&A. Diane, I think you wanted to say a few words at this point before we pivot to the Q&A.
No, sure. Thank you so much. What a great discussion today, and we certainly appreciate you sharing your insights into applied research. So as a reminder to participants, you can submit your questions to the Q&A feature, not the chat. We will try to get to as many questions as possible, including those that were submitted in advance. So Mark, I'm going to turn it back to you, and let's start our Q&A.
Okay. So there's a question coming from the Q&A to Dr. Moore, to Matt Moore. How many of your devices are currently in the field, and how many more would you expect to be introduced in the coming years?
Well, we're in the process still of doing the research to develop both of these devices. I think specifically this one is actually deployable in the field, the amplification technique. We don't have any commercial IP associated with it, so we've published sort of how to do that for just the broader community. But so if people do want to use it and adopt the technique, they can immediately adopt it. Our other nanopore sensing technique, we're working towards trying to eventually get to that point. But you know, in terms of research, it's always, you know, you have to face those challenges and try and optimize it a little bit more. So we're getting close, and that's the end goal. But we're still working on it right now, research-wise.
Okay, thank you. Phyllis, there was a question for you. What are the results of efficacy testing on the mask inserts? Will copper mesh work as a filter in a cloth mask?
I have 12 pages of information on efficacy testing. So just very briefly, some of the tests are, yes, the mask is biocompatible. That's one of the efficacy tests. It's non-flammable. That's another efficacy test. We did manage to get 90% kill when we inoculated a mesh mask with a saliva solution. It's called salivary challenge. So if anyone is interested in any particular efficacy test, they can call me and let me know. And I will be happy to share this information with you. And I do plan to get it up on the website. And you wanted to know if the insert could be used in a cloth mask. Yes, it can. The insert can be used, but be very careful. Do not put this insert into a pouch. And the reason being, this insert releases copper atoms. And the minute you put it into a pouch, you're liable to negate the efficacy of the copper mesh.
Okay, thank you. So here's a general question. And maybe both of you have some perspective on this. With a lot of people now working remotely or working with social distancing protocols, how has this impacted how people problem solve, and how have they mitigated it? Let's start with that one.
Who goes?
Whoever, if you have a perspective, Phyllis, that would be fine to start with you.
Well, actually, I work generally alone, and I'm used to it, and I like it. And my hubby's out right now, and when nobody's around me, I can get a lot done. I don't think I need to mitigate anything. It's really a delight to be able to do that and not get those constant interruptions that I did when I was working at the medical center. The downside of that is the interaction with people and the the rapport that you have and the sharing of ideas, because there's no denying, and I always told my staff this, the group brain is better than the individual brain anytime. And the best group is about 3 people. That's what we found. So it has its pros and cons. Mostly I really like working in a quiet environment.
Yeah, Matt?
Yeah, that's a great question. It is. It's definitely a little bit different. I think with Phyllis, because we can do a lot of just collaboration and electronic work remotely in terms of the day-to-day function, it maybe isn't quite as much of a challenge. But then when you start talking about, just the social aspect of, students getting to work with each other, especially our department is a pretty close-knit community, and so not seeing your friends or not being able to connect with your students is definitely a challenge, and to some degree it definitely can inhibit developing those relationships that you mentioned earlier that are really key to a lot of applied research, especially that's interdisciplinary, like in food science. So it definitely is a challenge. Our department is trying to think up and brainstorm this semester of doing some activities where we can engage graduate students and undergraduates, and our food science club, which is run by undergraduates, is also planning a few events. So we're trying to make it work, but it's definitely a challenge.
Yeah, it's certainly different. That social connection is really important to us, you know, as humans, but also, you know, to us creatively. I think, you know, I'm well known for like popping into a colleague's office or the students, you know, into the lab and saying, you know, having a question or seeing what's up, and those kind of quick, low barrier things are much harder to do when we're all working remotely. But what I was going to say is that, you know, science, fundamental science and applied research, these are creative activities, right? You know, we're inventing things, we're coming up with possible solutions, and that group interaction, Phyllis, I totally relate to what you're talking about with regards to this, you know, us being able to come up with solutions for us to be able to even form the questions. What are we asking the right questions is important. So, you know, do you think that remote working is stifling our creativity, and do you see ways around that possibly?
It can. It cuts both ways. It can, and then it can give you the downtime so that you can actually think of these things.
Mm-hmm.
And I should offer, Matt, if there are projects that you think related to the maths that your students would like to collaborate on, I'd be very happy to supply material for you.
Oh, that sounds great. Absolutely.
Yeah, so these types of things that UMass is doing, they're bringing all of us together. I would have never met you. I would have never met Mark or Diane. I'm pleased to meet you. And like I say, it's a nice opportunity. And the area I'm in is very fertile. I've been fortunate to be in this position before when we did our first research in 1983 on the brass and stainless steel doorknobs. To summarize that, the brass killed bacteria in 7 hours. We realized it was a copper in the brass that was creating this extensive cytal activity that killed microbes in less than 7 minutes. But what we found out, and it's not really published well, is that the stainless steel threw the organisms into stasis. And I can tell you because you're at UMass. But basically, the difference between being cytal and being static is if you're walking across the street and you get hit by a car, that's cytal, you're dead. But stasis is you're alive, but you're not multiplying. And what that does is that keeps the bacteria viable for months. And we did find that as a problem at the medical center. And you find multi-resistant organisms on a doorknob, then you find it in a septic patient. So those are the kinds of things that, you know, are pretty interesting.
Yeah, Matt, do you have any perspective, like, you know, has the remote situation hindered the dynamics of your group, or have you found ways around it so that you can remain as creative as possible?
Yeah, it's definitely created a challenge. Even like with you know, students connecting, like we have an interdisciplinary program called the MCB program, the Molecular and Cellular Biology program, and I know they canceled the retreat. They normally have a retreat, you know, in the beginning of the semester, the fall semester, and you know, everybody shares posters, and you have networking, and usually you can, you know, ideas can come out of there, and so things like that, missing that, I think really can hinder creativity. But you know, you still have to, you know, being remote, we've still tried to find ways of doing it, but it's definitely been a challenge, especially you know, interdisciplinarily, you know, reaching out beyond just our department. It's definitely taken a hit.
Yeah, and you know, I see that, you know, the pandemic hit us. We none of us were really expecting this, and we had to transition to a ultra safe mode very quickly. But now what I see is, from where I sit, is people starting to adapt. I mean, we'd love to be together in person, in close proximity and interacting, you know, sharing a whiteboard or a blackboard and et cetera. But I see things happening to take place of those interactions, like you mentioned, the MCB event where there's posters and so forth. People are creating ways of doing that safely remotely because that still allows us to be connected, perhaps not in the ideal way. And group meetings and using things like Slack to keep a group in constant contact with each other. You know, I see that we're sort of rising out of that initial restrictions that we had to have a lot more and more activity, which is really good. Matt, there's a question for you. I think it's for you. Can you share more about the fabrication of nanosensors?
Yeah, sure. So there's a whole slew or a whole bunch of different varieties. So specifically, the sensor that we're working on related to nanopore sensing, it utilizes a special protein that's been isolated from E. coli that's actually normally on the surface. And what these nanopore sensors are is essentially just a protein that has sort of a hollow sort of center. So it essentially forms a pore in the membrane of E. coli. And because of the very specific nature of this particular nanopore protein that we're using for sensing, it's very flexible. So it has like sort of a set of hinges on the outside. And when you have it with a hole in the membrane, you can actually measure ionic current across the membrane. And so the way it works is if you have a target on one of those hinges that can bind your virus or whatever microbial target you want, it'll change the conformation and disrupt the flow of ions across that membrane. And so you can measure that. And then one thing in particular about this pore that's really exciting is it has enough flexibility that you can generate a very specific fingerprint when you have something bind it. And from that very specific fingerprint, you can create sort of a database or a catalog that you can consult of different fingerprints. And that allows you that ability to determine which strain is binding as well as what organism is binding. And so it could be very, and so that's what we're trying to work on now, is developing and determining how we can best manipulate the pore and how it behaves best in sensing these viruses as well as bacteria and other pathogens of concern.
Thank you. Thank you. That's very interesting. Phyllis, there's a couple of questions for you. The first two are the copper masks available to purchase? Are the copper mask inserts commercially launched?
Yes, they are, and they can be purchased now on cubecoppersolutions.org, our website. Here, I'll advertise again. You got that?
Yeah.
Okay.
There's another question for you, Phyllis, is there a certain grade or specification of copper needed, or does all copper provide antimicrobial and antiviral benefits?
All copper does. All copper does, but we did a lot of research to get the screening exactly right. I need to add something, if you don't mind, because I think it will benefit our people. This is a regular surgical mask, and we've already shown it's impermeable to water. Now, a lot of people use these a few times and they throw these away. But what we found is if you take these, even though they're impermeable to water, and you put rubbing alcohol on them, it goes right through. And the reason is the surface tension is so much lower. So you can actually wet your whole mask and you can disinfect it. And then make sure that you dry it very, very well. And you've got a nice sanitized mask.
All right, thanks. Let's see. There's another question here. How will other projects and research at CNS work into the community around this pandemic?
Yeah, so actually, sort of getting back to the collaboration thing, the one thing that we did see about this pandemic is that we have a lot of scientists who are in the area of either immunology or virology, and actually having a common problem or a common thing to focus on has actually resulted in a lot of, potential new projects looking into SARS-CoV-2, of which our lab has been looped in on. And so there's all kinds of research, I'm sure, going on right now on SARS-CoV-2 related to campus. So just to sort of as an example, there's a really cool proposal that Barbara Osborne and a new faculty hire, Alyssa Rothschild, put in related to trying to come up with a sort of a better model for studying the immunological response that our bodies have to infection with SARS-CoV-2. There's, our lab is actually also trying to look into understanding viral transfer because, there's a lot of epidemiological evidence that surfaces don't really play a role very much, or a very heavy role in SARS-CoV-2 transmission. But if you have a mechanism to couple with that to really say with better confidence, hey, these don't really play as big of a role as we originally would have thought. It's, that's one of the things that we're actually working on. So Lynn McLansborough in our department actually has done a lot of research on bacterial transfer with a specialized transfer machine that can control for pressure, time of contact, things of that nature. And so one of the things that we want to do is truly empirically say, okay, if I have SARS-CoV-2 on a surface and I touch it, how much is actually going to transfer to my hand? And then from that, how much is actually going to transfer to my face, right? So just because you can identify SARS-CoV-2 on a surface doesn't necessarily mean that it's going to get into your mucosal membrane. There has to be a transfer event. And until you really understand the efficiency of that transfer, you don't necessarily have the most robust mechanism for understanding how the virus is transmitted. And so that's one of the things that we're looking into. So there's a whole, that's just 2 examples, but there's a whole bunch of projects that people across campus have been working on that's really exciting.
Yeah, I agree, Matt. I've seen a lot of grant proposals get funded for COVID-related work in the college, across the college, and in many different areas, like you mentioned, vet and animal science and food science and chemistry. I've seen the psychological and brain science. So I'd say in general, people see an opportunity to use their specific skills and experiences to contribute to the effort in new ways. There was also a question that came up, which maybe I'll answer here, but Matt, you can also answer. The question was, how would, we in the College of Natural Science vet proposed ideas to be accepted as future projects? Is there any process we follow? And we might hear of many candidate ideas. How do we select them? Well, I just say to the community in general is that we like to hear from industry. We like to hear from people in the community about potential ideas or opportunities and potentials for partnership. And what we do if we get these questions in the dean's office, we try to direct them to the right experts in the college. We have a broad portfolio of expertise. And oftentimes there's somebody who can speak to it. There's not always a possibility for a match, but many times there is. And we try to help when we can. And it always depends on the details, right? So a discussion is necessary as long as we get sort of the right people who might be able to be in potential partnership to discuss, then things can go forward from there. Matt, I don't know if from your perspective, if you have that can answer this vetting and the process of ideas.
Yeah, I agree completely with what you said. It's really kind of up to the idea itself, and then also if it's an idea for a collaboration or a project, getting the funding for it as well. So we can put in these grant proposals to NSF, but then on top of it, we actually need to get the grant to have money to actually do the research. So presumably, you want to put in something that's actually a real direct need and that you can make a really strong case, for conducting.
Yeah, I agree. It does take money to do research. We all know that. But yeah. And it's historically, it's come in different ways. Sometimes industry funds a specific project that's a partnership project. Sometimes it's money from a donor for research in a certain area. And sometimes it's, like you said, going into a proposal, sometimes a joint proposal towards the applied side of National Science Foundation or one of the other agencies to have things go forward. I see we're at time to transition. So before we turn it over to Diane to close, I want to thank you, Matt and Phyllis, very much for the interesting discussion and telling us about your work. It's great to hear of all these exciting ideas and the progress that goes along with it. So I'll turn it over to Diane.
Wonderful. Thank you so very much. Our time has concluded today. And again, thank you, Dr. Tuominen, Dr. Kuhn, and Dr. Moore. What a great opportunity to hear from you. To our participants, thank you for joining us. You will receive a follow-up email survey. We hope you'll take a few minutes to complete it and share your feedback and ideas for future webinars. There are questions that we were not able to answer during today's session, and we will try to post some of those questions as well for a follow-up. Please be sure to follow us on the UMass College of Natural Sciences social media to stay updated on news and future events. Thank you again. Continue to stay well, and we'll see you at the next webinar.
Thank you.
Thank you.
Thank you.
Have a good one.